Hello everybody, welcome to today's session and the chapter is Human Health and Diseases. So last class we were on session 1 and we had discussed about certain different types of pathogens which cause diseases to our body. We have learnt about health, how we can prevent or how we can maintain a balanced diet, how we have to do regular exercise to maintain or to boost our immunity as well as lead a healthy life. In last class we have discussed about disease which are infectious and non-infectious. We have come across pathogens and these pathogens were of different forms like bacteria, virus, helminthes, filarial worms, protozoans, etc. In last class we did study about different forms of bacteria, that is which causes typhoid as well as pneumonia, and we have come across virus, that is rhinovirus, which causes common cold. One of the important thing what we had discussed last class was about protozoan, that is disease was malaria and the protozoan was Plasmodium vivax. We have learnt last class about life cycle of Plasmodium vivax. So in today's class we will be initiating the class with one more type of protozoan and the disease here will be amoebiasis, or it is also called amoebic dysentery. So this amoebiasis or amoebic dysentery, it is caused mainly due to a protozoan and the protozoan is Entamoeba histolytica. Entamoeba histolytica, it is a protozoan which causes this amoebic dysentery or amoebiasis. So when it comes to effect of or the symptoms of Entamoeba histolytica, this normally affects the large intestine. So in the large intestine, what is the function of large intestine? Large intestine mainly helps in absorption of water and release of the excretory products. So when it comes to this Entamoeba histolytica, it initially affects the large intestine and you might wonder how does it travel inside. So here there are vectors and the vector here is a housefly. So along with the fecal matter of an infected person, these Entamoeba histolytica will be removed. So if a housefly sits on this fecal matter and sits on any food or drink what you consume and contaminates it, then there are chances that Entamoeba histolytica normally can be transferred into the healthy human. So housefly acts as a vector here. Once the large intestine is affected by Entamoeba histolytica, it interrupts in the normal functioning of large intestine. It doesn't allow absorption process, so the stools will be watery, that is fecal particles which is removed from the body will be liquefied and it is rich in or it is very much high in mucus. Plus there will be certain blood clots, which that is mucus and blood will be released along with the fecal matter. Apart from that, constipation, stomach pain, abdominal pain, these are the common symptoms of amoebic dysentery or amoebiasis. So amoebiasis is caused by a protozoan Entamoeba histolytica and the vector here will be a housefly. Vector is nothing but the carrier which carries the protozoan into a healthy person's body. So that is about amoebiasis or amoebic dysentery. Next we have to learn about helminthic worms. So helminthic worms, there are two different forms what you will be learning. One is Ascariasis. Ascariasis is the disease which is caused by Ascaris. Ascaris is a helminthic worm and the scientific name of Ascaris is Ascaris lumbricoides. Ascaris lumbricoides is a helminthic worm which causes the disease Ascariasis. This Ascaris lumbricoides enters into the human body, especially you can find them largely in kids. Why kids? Because they are also released along with the fecal matter and they get mixed up with the soil. So the eggs will be present in this soil. So children normally play in mud and the same hand will be transferred into their mouth. When they transfer the mud filled hand into their mouth, there are chances that the eggs of Ascaris lumbricoides might get transferred into their body. So this is a type of endoparasite which enters into the body. Their location or where they live, that will be the intestine. So intestine means again here they will be interrupting the absorption process. So in case if their number is very much high or the eggs are very much large, then it leads to intestinal or stomach pain. So commonly we call it ಹೊಟ್ಟೆಯಲ್ಲಿ ಹುಳ ಆಗುವಂಥದ್ದು. So this hula is nothing but Ascaris lumbricoides. These have a sexual dimorphism, that means male and female can be clearly identified. As they are endoparasite, they have protective measures against our intestinal juices, stomach juices to protect themselves. This Ascaris lumbricoides, once their number increases, if at all they are not treated, then there are chances that they might travel into the upper part or the above the abdominal part of human body and may be released from other openings like nasal cavity and all. So that is why once their number is increased, they will start to reduce the absorption, so automatically there will be loss of appetite. Intestine process or absorption will be altered as well as appetite will be lost. Constipation will be there as well as certain severe cases, certain types of vomiting also will be present. So next helminthic worm that will be, or the disease is Phylariasis, or it is commonly called Phylariasis or elephantiasis. So Phylariasis or elephantiasis, as the name suggests, here it is caused because of a particular organism, that is helminthic worm, and that is commonly called filarial worm, or the scientific name for this will be Vaucheria bancrofti. Vaucheria bancrofti is one of the organism or helminthic worm or filarial worm which causes this Phylariasis. There is one more species that is called Vaucheria malai. So two helminthic worms or two different species of helminthic worms, that is Vaucheria bancrofti and Vaucheria malai. These are the two organisms which leads to Phylariasis and elephantiasis. So these have a vector that carries the organism or the worm into the body of the organism, that will be mosquito. So the vector here is a mosquito. Now here it is not Anopheles, sometimes it will be Anopheles as well as Culex. These mosquito, what they do is they affect, you might have seen that the mosquitoes enter into the house somewhere around 7 or 8 o'clock, but they will be simply roaming around and they bite the individual only somewhere around 1 o'clock or 2 o'clock. Why? Because when the individual is in its deep sleep, so that their chances of them getting killed is very much less. That is why these mosquitoes, so normally in any person who is affected by this elephantiasis, his particular filarial worm will come just below the skin somewhere around 2 AM in the morning. So what happens? The mosquitoes will bite this individual who is affected by this filarial worm and takes up the worms into their salivary glands. When the same mosquito bites a healthy individual, these filarial worms will be transferred into the body of this healthy individual. So all these activity takes place somewhere in the 1 o'clock or 2 o'clock range, that is in the very early morning. So once this filarial worms enters into the body, first what they do is they affect the lymphatic vessels or the lymph vessels. Once they start, what do you mean by they attacking the lymph vessel is they start to reduce the activity of the lymphatic system. Apart from this one, their number increases in such a range that they start blocking the lymphatic vessels. Due to the blocks in the lymphatic vessels, normally our abdominal part of the body will start to swell up, that is the lymphatic swelling will be there. Because of this lymphatic swelling, there will be increase in the size of the locomotory organs, especially in the affected individual. So what happens, that is the one which gives the name elephantiasis, that means Anekalu Roga, because of which one there will be increase in the size of your legs. But here it does not affect only the locomotory organs, still in certain cases it even affects the genital organs also in case of males as well as female. So this is an irreversible change. So once if the filarial worm enters into the body, it affects the lymphatic system, the size of the locomotory organs it increases. What happens here is there will be no reversible change. That means if the size of the leg increases like this, and that is because of the swelling in the lymphatic vessel. Once the lymphatic vessel, or once we identify the symptoms, we can give the treatment. Further damage to the lymphatic vessels can be treated, but this increase cannot be reversed back. Once increased size cannot be reversed back and brought back to normal. It is a permanent change that is there in your body. So that is about Phylariasis or elephantiasis, which is caused by Vaucheria bancrofti or Vaucheria malayi. Here the vector is a mosquito. So that is about helminthic effect or helminthic worm. Next one will be a fungal disease which is called ringworm disease. This ringworm disease is caused by or caused because of certain types of fungi. They belong to three different forms of genus. One is Trichoderma, another is Microsporum, and one more will be Epidermophyton. So these are the three different genus to which the fungi belong. They belong to different species of these three genus and causes ringworm disease. Now what is a ringworm disease? What happens? So these fungus can develop easily when the temperature is having heat, that is heat and moisture helps in increasing the development of fungi. So you can see this fungi especially in the toe, that is between the fingers especially in the legs or in the hands. If they start to increase, then it leads to continuous itching. There will be continuous itching which normally causes skin rashes. Certain types of lesions will be produced. Lesions means gulegalu. So these lesions will contain mucus or watery component which increases the lesions. So what can or how can we prevent this one is you have to have proper bath. Every day clean yourselves, proper bath will normally remove this fungus from the body. So it is again a spreading disease. So how can it spread anta kedadre? These normally spread if you use the same utensils used by a infected person, especially comb that is used by a infected person. If you use the same comb, then what happens? This fungus gets transferred into your scalp and there will be itching in your head. So there are chances that the infected person unknowingly might transfer the fungus into your body. So this fungus affects the beard region, neck region, toe region, leg region, armpits region, where the amount of heat and moisture will be very much high. So here these are about different types of diseases caused by different forms of pathogen. So once again to discuss, today we started the class with amoebiosis or amoebic dysentery. Here it is an example or the organism or pathogen causing this amoebiosis or amoebic dysentery. that will be an example for protozoan. The protozoan here will be Entamoeba histolytica. So this Entamoeba histolytica is the one which causes amoebiosis or amoebic dysentery. Here the vector will be housefly, which transfer this protozoan into a water as well as food. That means it contaminates the food or water by transferring. The housefly acts as a vector here. Next one is two different forms of helminthic worms. So here it is Ascariasis is a disease and Phylariasis or elephantiasis is a disease. Ascariasis is caused because of a helminthic worm, that will be Ascaris lumbricoides, and Phylariasis or elephantiasis is caused by two different forms or two different species, that will be Vaucheria bancrofti and Vaucheria malai. So these two causes the Phylariasis or elephantiasis, where mosquito acts as a vector. Next one is about fungus, that is ringworm disease. So ringworm is the common name which is given. So you have to write ringworm disease. So ringworm disease is caused either because of Trichoderma, Microsporum, Epidermophyton. So these are the three different genus to which the fungus or to which a particular species belongs, which is responsible for cause of this ringworm disease. So when it comes to this ringworm disease, it causes lesions, itching because of the water component which is present within the lesions, and will lead to continuous itching and scaly skin, or even it makes rashes all over your skin because of the effect of this fungi. So it is having a very easy, rather than cure, you can tell it is prevention. So if you keep your body clean, that is bathing regularly, cleanly maintaining the body, then it is very easy to avoid this fungus. So that is about different forms of pathogens which affect our body. Next you have to study about different forms of prevention method. So next is about prevention. Okay, so what is prevention? You might have heard that prevention is better than cure. That means rather getting subjected to a particular pathogen, getting that disease, expressing the symptoms, and then finding a solution for that is a very long process and very much dangerous to your body. Rather than that, if you are able to prevent your body from getting exposed to that kind of pathogen or disease, it will be very easy. So here it means prevention means protecting your body from or against any of the pathogens or foreign substances attacking your body. So how can we maintain our hygiene? That means there will be two types of hygiene, that is personal hygiene and public hygiene. So personal hygiene you can easily maintain by taking regular baths, maintaining your body clean, especially between the or in the nails, cutting them properly, or if not, cleanly maintaining them. So everything, washing your hands properly, if you go outside, washing your legs and hands properly before you take food. All these are the general basic hygiene which an individual can maintain himself to protect against any of the pathogen attack. Now what do you mean by public hygiene? So public hygiene here is not only your responsibility but a responsibility of a particular organization or a particular management as well as the government. That means if there are any places like for example bus stands and all, it should be cleanly maintained. If any people who find, or you might find any particular people spitting all over, then it is once again a reason. That means there are chances that his body or spit might contain certain types of pathogen which might affect the individual, or it might get transferred through aerosols and might affect the healthy individual. Like bus stands, hotels, swimming pools, cesspools, tanks, everything should be maintained in a clean form or a pure form. If you see malaria, we had discussed about malaria the life cycle of Plasmodium we had discussed. So in this one you might have seen that Anopheles mosquito is the one which is the reason for, or this is the vector which causes, or you can also see housefly in case of amoebiosis or amoebic dysentery, mosquitoes again in case of elephantiasis. All these are the vectors or the carriers which carry this disease from one individual to another one. So what we have to do is we have to reduce the breeding of these organisms. Their breeding ground should be initially destroyed. You know that mosquitoes reproduce and lay their egg in the pure water, and the larva will be developed in the pure water. So what we can do is we can either properly close the pure water, like smaller tanks in your house that can be properly closed. Apart from this one, if you see larger swimming pools, ponds and all, what we can do, I had told that we can introduce a fish there, and the name of that fish is Gambusia. Gambusia is the fish which consumes the mosquito larvae for food, okay, which reduces their population, and the chances of them spreading the disease is very less. Even housefly also, if there are any excreta, dog or cat or any human excreta, which is very common thing which normally transfers or makes the vector housefly to transfer the components into or the pathogens into the human body. So that can be cleanly removed by adding mud over it, so that none of the mosquitoes or houseflies sit on that. Or else again the breeding grounds of these houseflies or mosquitoes should be destroyed. That is one way. Apart from this one, chikungunya, dengue, this is formed because of a mosquito, and that mosquito is Aedes. Aedes is the mosquito which causes this disease, dengue as well as chikungunya. Again here also the only reason or only way to make this disease come into control is by avoiding the breeding grounds. That means destroying or avoiding the breeding grounds can be done. So the vectors we have to identify, their breeding ground should be destroyed, automatically their population decreases as well as the spreading of the disease. One more concept is about immunization and vaccination. Immunity and vaccine. Okay, there are certain different forms of diseases which have been completely eradicated, like smallpox, which is declared by WHO that it is completely removed from the earth. So before we know that what is a disease, we should identify what is the reason behind the cause of this disease. Once we identify the reason, we have to identify the characters of it, how does it spreads the disease, what is the way our body reacts to that type of organism or pathogen, or what are the symptoms. Once everything is identified, then only we have to identify or we have to produce a particular antibody or antigen, or in short medicines for that particular pathogen. So naturally our body is the one which protects us from certain types of pathogen, but in certain cases our body fails to do that. That is why what happens is our body should have some excess or some additional forms to destroy this, that will be given by vaccines. So in together we have to undergo prevention both personally as well as in public, that is hygiene should be maintained both in public and private, that is in person, so to avoid such kind of diseases. So that's a short note on prevention. Next we will discuss about immunity. Immunity means it is the ability of our body to protect our body against disease causing organisms, that is pathogen. Any pathogen which enters our body will not successfully stay in our body or cause disease to us. But there are certain ways the immunity protects our body to avoid the pathogen from entering. If at all entered, it gets destroyed within the body. So immunity means which protects our body against certain pathogens. Now immunity, there are two different types, that is innate immunity and acquired immunity. Innate immunity is the one which we have got by birth, that means it is inherited from our parents to us. That is what is termed innate immunity. So these innate immunity acts in the same way to any of the pathogen which enters our body. Innate immunity has certain forms of barriers which protects our body from different types of pathogens. So we will study about the barriers which protects us from pathogens. So innate immunity means these are the immunity which is inherited by birth. These contain certain forms of barriers. So here it will be just a minute. Physical, physiological, cellular, cytokinins. There are 4 different forms of barriers: physical, physiological, cellular and cytokinin. Now what is included in physical barriers? Physical barriers means the outermost protection what is present in your body, that will be your skin. So skin forms the physical barriers, along with which mucus which is present in GI tract, that is the gastrointestinal tract or your digestive system. These both form the physical barriers and do not allow any and every pathogen to enter inside your body. So next one is physiological barriers. When it comes to physiological barriers, even though the pathogen passes this physical barrier, they have to face the second barrier, that will be physiological barrier. So physiological barrier, if something enters into your eyes, what happens? Water will be released from the eyes, or tears will be released from the eyes. These tears have higher amount of salt concentration which do not allow this pathogens to enter inside, and that will be shedded off from the body. Apart from this, in your digestive system, that is the presence of concentrated HCl which destroys this pathogen. So these are the components, either mucus, either tears or acids, digestive juices, intestinal juices, all form the physiological barriers which protect your body from pathogen attack. Next one is the cellular barrier. So cellular barrier includes all the defensive cells, that is WBC cells, neutrophils, eosinophils, basophils, monocytes, lymphocytes, B lymphocytes, T lymphocytes, natural killers, mast cells, PMNLO. All these organisms or all these cells destroy the pathogenic organisms and protect our body. So next one is cytokinins. So cytokinins are particular forms, that means if at all, for example, if this is the cell present in our body, this is affected by a virus. So once a single cell is affected by a virus, the nearby cell are healthy cells. This, if affected by virus, these starts to release a particular type of chemical, and that chemical is interferon. These interferons are released by virus affected cells. What is the role they play is, these interferons go and reach the healthy cells, so to indicate or make this cell prepared against certain forms of virus, so that that can be destroyed. So interferons are released, or these are the chemicals which are released from the virus affected cells, which just is the indication, or it just goes and indicates or intimates all the cells, so that it will be ready with the antibodies to destroy this virus. So that comes as a barrier in cytokinins, and it is restricted only for virus affected cells, not to bacteria or fungus affected cells. So these are the different forms of innate barriers. A very important 3 or 5 mark question. Innate means immunity with which we are inherited, that is got from our parents. They include different forms of barriers, that will be physical barrier, physiological barrier, cellular barrier and cytokinins. When it comes to physical barrier, it includes the skin and mucus present in the GI tract. Physiological barrier, it includes different forms of digestive juices, it includes concentrated HCl, it includes the intestinal juices, enzymes, everything come under physiological barrier. Next one is cellular barrier, which includes our defensive cells, that is most of the forms of WBC cells and lymph cells. So in together these protect our, these are the third layer of our protection. Last layer will be cytokinins, but these are restricted to only virus. If any virus affects the individual or healthy cell, they release a chemical termed interferons, which intimates other cell to produce the antibodies and to destroy the virus. That is how innate immunity is having different forms of barriers. Next one is, or next form of immunity, that will be acquired immunity. So acquired immunity means these are pathogen specific. These are pathogen specific. So pathogen specific means based on what type of pathogen has entered our body, the acquired immunity gets activated and their response will be different for different forms of pathogen. A virus attacks our body, the way of attacking or reverse attacking will be very much different from the bacteria or the fungi. So when it comes to acquired immunity, there are response towards a particular pathogen. So first response which is provided by our immunity towards the pathogen for its first encounter. ಮೊದಲನೇ ಸರಿ pathogen ನಮ್ಮ ಬಾಡಿಗೆ ಎಫೆಕ್ಟ್ ಮಾಡಿದಾಗ the ability of our acquired immunity to respond to that particular pathogen, that is termed primary response. So primary response will be a little bit weak, okay, because as the pathogen is entering for the first time, and first our immune cells should identify what is the type or what is the way or form of the effect of this particular pathogen on our body, and then start to identify and destroy this particular pathogen. So it takes a little bit of time, and that particular form is termed primary response. This will be a little bit weak. If the same pathogen enters to our body frequently, continuous, if our body is exposed to this kind of pathogen, then the response is termed secondary response. And this secondary response, that will be very much quick, because we already know the pathogen, we know how it is, what is the structure, what is its effect, and we already have certain types of antibodies to destroy that. That is what is called a secondary response. So primary response is the first response for the pathogen encounter for the first time, and secondary means frequent visit of this pathogen to the body. The response produced by this immunity or acquired immunity is called the secondary response. Now in case of this primary and secondary response, there are different forms of cells involved. The cells involved in this particular are B cells and T cells, that is B lymphocytes and T lymphocytes. Now for example, primary and secondary response, it is mainly because of two different types of cells, that is B lymphocyte and T lymphocyte. So B lymphocyte means it is released from bone marrow, that is why it is termed B lymphocyte. T lymphocytes are released from the organ thymus, that is why it is termed T lymphocyte. How do the, or what is the effect of these cells on the pathogen? If you ask, the B cell produces large, if any pathogen enters into the body, then the B cell produces large or army of proteins. They release higher amount of proteins into the body fluid, that is the blood. So these proteins are nothing but antibody. The proteins are which are released from the B lymphocytes will be antibodies. The growth and development of these antibodies, it is controlled by T lymphocytes. So B and T lymphocytes are produced in response to primary and secondary response. These B lymphocytes release the army of proteins, and the proteins are termed antibodies. The growth and development of this antibodies is controlled by T lymphocyte. Now we have to know the structure of an antibody. So we will draw the diagram of an antibody. This is a very important 3-mark question. Structure of the antibody will be asked in your examination. So this is the diagram of a antibody. So antibody, it contains, or it is a protein, as I have already told, it contains carbon and nitrogen. So when it comes to this one, here the structure of this contains two different chains, two different types of chains, light chain and heavy chain. In together there are 4 chains, that is 2 light chain and 2 heavy chain. That is why it is called L2 and H2. The structure represents L2 and H2. So L1, L2, this is H1, H2. So these are light chains or heavy chains, are held together with the help of sulfides or sulfide bonds. At the end of a light chain or a heavy chain, there is a particular part which is called antigen binding site. This antigen binding site, it is present on either sides. This antigen binding site is different for different pathogen. So you can see here the serrations. These serrations vary based on the type of pathogen it encounters with. So if a pathogen comes here and it matches with this antigen, then the pathogen or the antigen will be destroyed by this particular part of antigen binding site of a antibody. So this antigen binding site is different for different types of antibodies. So the structure here contains nitrogen at the antigen binding site and carbon at the base of the heavy chain. So there are 4 chains, light chain and heavy chain, 2 light chains and 2 heavy chains. These 2 light chains and 2 heavy chains are bound together or held together with the help of a sulfide bond. So antigen binding site varies based on the pathogen it encounters with. Based on the presence of the antibody released by our immunity, the acquired immunity can be classified into 2 different forms. One is humoral immunity. It is purely based on the types of antibodies produced, and one more is cell mediated immunity. This is also called CMI, that is the cell mediated immunity or humoral immunity. So humoral immunity means it is based on the type of antibody released, and cell mediated immunity means here it is restricted, type of pathogen enters and then the cells mediate the immunity. That is how it is classified into humoral and cell mediated immunity. These are the types of acquired immunity. So once again, to discuss about the immunity. So immunity means the ability of our body to reduce the effect or to prevent our body from the pathogen attack. So immunity are of 2 different types, that is innate immunity and acquired immunity. Innate, with which we are born, and acquired means we learn this. That means based on the pathogen we encounter, there are different forms of antibodies produced in our body. Innate immunity have 4 different barriers: physical, physiological, cellular and cytokinins. Whereas acquired immunity, they have 2 different forms of response, that is primary response and secondary response. Based on the first encounter, it is primary. If the repetitive encounter of the same pathogen takes place, then the type of response is secondary response. These 2 release, for their immunity purpose, they release 2 different types of cells, that is B lymphocyte released from bone marrow, T lymphocyte released from thymus. The B lymphocytes produces or releases a large amount of protein to destroy this pathogens. These proteins are nothing but antibodies. The development and growth is controlled by the T lymphocytes. So this antibody has a particular Y-shaped structure with light and heavy chains and an antigen binding site. Based on the structure of this antigen binding site, there are different forms of antibodies. If you see the different forms, some examples will be IgA, IgM, IgG. These are the different types of antibodies. Why we have classified it into different types? Because they are different based on their antigen binding site. So apart from this one, they have sulfur bonds which holds together the light and heavy chain together. Once these antibodies are released, because of presence of these antibodies, acquired immunity is divided into 2 different forms. Presence of antibodies, it is called humoral immunity, and if the immunity is maintained by a particular forms of cells or groups of cells that mediate the immunity, then it is called cell mediated immunity. In next class, we will be learning about different forms of immunity, that is active immunity as well as passive immunity. Thank you.